You didn't mention that your macro special meeting is free booze.
Oh yeah, there's free drink at the macro special meeting.
Yeah.
Which we maybe shouldn't be advertising. All right, so our first speaker this afternoon is Gary Cowney, who is a legend. He asked me to say that. And he's going to talk about patterns of change in the pollinators of Britain.
Great. So yeah, I'm just going to give you an overview of some work we've been doing at CEH, looking at trends in pollinators. So I don't need to tell you why pollinators are important, but despite all the research into pollinators, we still lack national scale species-specific estimates of change. So what I wanted to do was analyse all of the biological record centre data we have to essentially estimate these species-specific trends. So just as a quick overview of the type of data I'm dealing with here, it's biological records, so it's what, where, when type data. So it's, what species, where it was recorded, so this is usually gridded type of data. So everything I'm talking about is actually at the 1K scale. And when, a date, what date was it recorded. So it's usually collected by volunteers, like these little chaps along here. It's often collated and cleaned by various schemes and societies, and then it's made available for our use through things like the Biological Record Centre, NBN, GBIF, places like that. But the reason that this type of thing hasn't really been done too much before is that there's a lot of biases and problems when trying to estimate reliable trends from biological records. So this is one key example of these issues. So we've got year on the x-axis, number of records on the y-axis. The y-axis has been logged as well. So these are huge exponential increases in the number of records we've got across lots of different taxonomic groups. So obviously that inhibits our ability to estimate reliable trends. Other things that can cause problems are things like the publication of a new field guide or new ID keys, that type of thing. That can stimulate recording of certain species, certain groups, again inhibiting our ability to detect reliable trend estimates. So it's not all bad news. There's lots of solutions through the scientific literature, and they're all nicely summarised by some colleagues of mine, one of which is in the audience, where basically we're now looking at Bayesian occupancy models as the way forward. So this is a simulations type study, looked at all the various forms of bias associated with biological records, and Bayesian occupancy models tend to perform the best at extracting these reliable trends. And now the literature is generally kind of heading that way as well. Lots of recent papers have been using these models. You'll be pleased to hear that I'm not going to go into any details of the models, but if you want to chat to me afterwards, I'll make sure I'm around so we can have a chat then. But essentially this is the key output from the occupancy models. So it's essentially a time series of the proportion of occupied sites. So you've got occupancy on your y-axis here, year again on the x-axis. And as this is a Bayesian approach, this is a summarized posterior distribution. So you've got the median, which is the black dots, and then the 90% credible interval surrounding that, representing the uncertainty in the estimates. So I ran this for over 500 species of hoverfly and bee in Britain. So it's based on over 400,000 biological records. And these are just a few examples, example outputs for different species. We've got a couple of species that are decreasing at the top there. But what these nicely highlight is how Bayesian occupancy models give you this sort of a good estimate of uncertainty. So at the start of the time series, we obviously have less data, so that uncertainty is quite nicely represented here. And additionally, you'll see at the end of quite a few of these, the uncertainty begins to widen out again, and that's simply a result of a lag of getting the data from the recorders and from the schemes into the data that we're analysing. So for this analysis, I dropped various different species following a few different exclusion criteria. The first of which was to drop species that are not effective pollinators. So things like this high layer species up here, these tend to take the pollen, clean it off themselves, and sort of take it down into their crops, so they don't actually act as effective pollinators. So they're out. Things like cuckoo bees are gone as well. And we also dropped species where there's been known taxonomic issues, and also species where the models just didn't perform particularly well, so where several of the parameters didn't converge or the uncertainty estimates are just completely wild. So using these data, I wanted to address these three following questions. I wanted to try and figure out what's happened to our pollinators over the last 30 years. I wanted to know if there's any interesting patterns in the trends, and I also wanted to think about the potential implications of these trends for pollination services. So first things first, we need to extract a trend measure from these outputs. So this was done quite simply as the proportion change between 1980 and 2010. So again, this is a Bayesian approach, so we've got a posterior of estimates of this change. So we just take the median here and then the 90% credible intervals of that change. And we chose 2010, not 2013, because of the what I was telling you earlier about the uncertainty in the final few years. So in terms of question 1, overall we found that there was a 15% decline across all species and across all of their posterior iterations, which is shown on this graph here. So this is essentially a density plot, where it's essentially like a smooth histogram. So we've got trend on the x-axis here, and the solid line along here is the 15% decline across all species, and then I've separately plotted bees and hoverflies here, and you can see that they're roughly declining by a similar percent as well. An interesting thing to note here is that it seems to be a lot of bees that are doing quite well on the right-hand side here. So this is exactly the same data, but plotted a different way. So each one of these points is a species, along with its uncertainty, again with its 90% credible uncertainty, which is on the y-axis here. I've just ordered them in terms of median, in terms of their trend, so the ones that are decreasing are this side and the increasing ones are that side. If we think back to the other plot, you can see that the bees on the far right, there's quite a few bees that are doing quite well, which corresponds to that increase on the other side, on the other plot. And the nice thing about this approach is that you can classify species as either increasing or decreasing based on whether their confidence limits band or span 0. So using this approach, we found that 12% of bees were increasing, 29% were decreasing, bees and hoverflies were decreasing, and then we couldn't reject sort of the null hypothesis, so essentially the remaining ones we're suggesting are, well, stable. So we just pick out a couple of examples. We've got a species that's doing quite well on the right-hand side here, so Andrena nitida. It's got a 41% increase, and we can contrast that to another species in the same genus, which is having a 40% decline, so it's the one to the left of this one with a huge uncertainty here. So what this doesn't tell us, those previous plots, is anything to do with any temporal patterns in these changes. So did these two 15% essentially come about from the same process to get to that same point? So I've just plotted mean occupancy here against year across all the species within the two different groups, and you can see that there's different patterns, different temporal trends to what's going on. So the hoverflies have shown this sort of gradual decline, sort of starting in the late 1980s, whereas bees, if anything, were kind of increasing sort of the mid-90s to the mid sort of 2000s, but then their declines are driven by these three consecutive bad years in the late 2000s. And on the train up here, it dawned on me, it almost looks like they're sort of fluctuating asynchronously, particularly in the end here. So it's almost like a good year for bees, a bad year for hoverflies. So just to try and get a quick look and a quick handle on that, I plotted the first derivative of one against the other, which is what's on the right-hand side here. So each one of these points is a year. It's essentially a measure of was it a good or a bad year for that group. And sure enough, they were negatively associated. So it looks like a good year for bees is bad for hoverflies and vice versa. But this has lots of uncertainty in here, which I'm ignoring. So it's only just it's just interesting, really. So the final result is it's in terms of the implications for pollination services. So to try and get a handle on this, I simply made the assumption that the widespread species are the species that we should be concerned about in terms of pollination services, because there's the ones that we have the most of them, so they're likely the ones that are doing all the work. So that's things like Bombus pascorum, Bombus lapidarius. So to just get a quick handle on that, I want to see if range size was associated with trend. And as you can see, there's nothing really going on here. But to take that one step further, I went to the literature and picked out some key sort of dominant crop pollinators in Britain. So there's 24 species, and that's the red line. This is their mean occupancy plot here. So if anything, these species look like they're doing quite well, which I guess is good news. But then if you contrast it to all the remaining bees, you can see that they've still shown this sort of a little bit of a decline there. So just to round things up, how our bees, I'll link back to the original questions. So in terms of how our bees are doing, over the last 30 years, we've seen a 15% decline on average across species, and we found that 29% of species showed significant declines. If we think about the sort of long-term trends and looking at how they vary between groups, you can see that we've had this sort of slow, gradual decline of hoverflies compared to the declines which are driven by declines in bees, which are driven by these 3 really bad years in the late 2000s. And finally, in terms of implications for pollination services, it actually doesn't look too bad because we've got these 24 species that are doing, okay, the ones that are thought to be the key crop pollinators. But obviously, I'm not saying that all's fine in the world because our dominant crop pollinators are doing well. You obviously don't want to put all your eggs in 24 species baskets. So, that's everything. So, thanks, and I'm happy to answer any questions.
Plenty of time for questions. Have you got any questions? Tom?
Do you know what was bad about those 3 bad years?
I've only had a little bit of time to speculate on it, and I've been speaking to Bee Wars. So this is the Bees, Wasps and Ants Recording Society, who all the data come from. And they kind of think that it might be driven by climate. So it could be just 3 particularly bad years for bees. So they also thought that a bad year for bees are when it's really damp type summers, and they sort of benefit from warm, nice sunny summers, and that kind of matches in with the good year, bad year hoverfly trend, in that potentially those really bad years for the hoverflies are when it's really nice and sunny, but because the larvae are exposed in the hoverflies, they're sort of more vulnerable to desiccation compared to bees whose larvae are in the nest. So potentially it could be something to do with climate. I try to avoid any speculation on what's driving bee declines for obvious reasons. But I guess if you wanted to know that, you should have gone to Nick's talk.
I guess relating it to pollinator services makes the assumption that the abundance relates to the occurrence. Do you think that's reasonable for the species?
Yeah, so the species that I picked out, the dominant crop pollinators, are kind of the widest, a lot of them are wider spread species. That final trend is the just mean trend across all occupancy. So you can see the intercepts a lot higher here. So these are generally the wider spread species. I've not really got any data on abundance per se, but if we manage to fund a pollinator monitoring scheme, then I guess that's the sort of question that will be directly able to answer.
Did you look at how the window of time that you're looking at affects the overall trends when aggregated? So if you pick different start and finish years.
I've not done any of that. I did think that's potentially a good way to go. So I did do some stuff. So when I was talking about how you estimate the trends, so we're just looking at the proportional difference between the first year and 2010. We looked at lots of different approaches there, fitting smooth lines and also fitting piecewise regression. So multiple ones of those. And they were all basically coming up with those, the breaking point was all just before those last 3 bad years. It was quite difficult choosing which way to present these results. And that was one of them. So maybe I should have gone with that.
Very quick last question.
Could there be any kind of risk of shifting baseline syndrome? So could we say everything's increasing, but actually it's experienced quite a lot of historic decline?
So if you look at the wider literature on this, a lot of people are saying that the declines happened sort of pre-1990, 1980, which was before what we've shown here. So as with any of these trends that are based in 1980 onwards, like the stuff Charlie presented earlier on the state of nature, well, that's 1970. There is an issue of shifting baseline syndrome. It's nothing we can really get a grasp of yet. If the data was there, it'd be better.
Okay, let's thank Gary again. All the people who've come in since I was started, if you're tweeting, if you could add the hashtag #VSMacro, that would help us curate things afterwards. That'd be okay.
Just wait for it to turn.
Thank you. Hi, so I'm Helen, and I'm going to be sort of carrying on a little bit about the environmental traits and talk about some work that I've been doing with the Landscape Ecology and Primatology group at Bournemouth in predicting, using biological traits and the environment in predicting species occurrence throughout Africa for diurnal African primates. So what we were aiming to do is to identify which biological traits and which environmental variables are most important in determining the occurrence of different primate genera across sub-Saharan Africa. The reasons we want to do this is, as we've already heard, we know that climate change and anthropogenic disturbance is going to significantly alter the natural environment, and species are going to have to adapt to this change. And if we can understand what's driving species distributions, we can predict, in theory, how they're going to change their range in response to environmental change, and this can help us to inform conservation policy. This is really important for primates because they're a very arboreal group of animals, so they rely on forest, and this is going to be significantly affected by climate change. And they're actually one of the most threatened of the mammal orders. There's 437 species listed on the IUCN Red List. 266 of those have a threatened status, so this is either critically endangered, endangered, or vulnerable. That's about 61% of the order, so that's quite a high proportion of that order that's classed as vulnerable. This is based on the idea of environmental trait filtering. So this is basically the idea that the biological traits of a species are going to determine, and along with the environment, are going to determine the habitat suitability, and that's going to impact on their ability to survive in a given habitat, which is going to determine their range. So in other words, because we predict that species are adapted to live within their particular environments, there's only certain environmental parameters that they can live in, so there'll be a relationship between the biological traits and the environmental variables that they live within. The methods that we use is we collected presence-absence data for 354 sites across sub-Saharan Africa, which you can see here. All of the black dots are our study sites that we used. The map here, this is a canopy height map of Africa, so the intensity of green shows the canopy height, so the darker the green, the taller the canopy height. We had 14 different primate genera that we were looking at, and all of these sites contained at least one of these different genera. These are the genera that we were using. So there's the 3 colobine genera. This is a colobus monkey here. This is a mantled goretza. They're arboreal, medium-sized leaf-eating monkeys. And we've got the Cercopithecines, this is a Sykes monkey. They're quite a diverse group of primates. They're generally smaller though, but there's a mixture of arboreal and terrestrial species in there and various different diets. Then we've got the Papionini, this is a olive baboon. So this is the baboons and the Galada baboons, the mandrills and drills, and the mangabeys. And then finally we've got the apes. So we've got Pan, which is chimpanzees and bonobos, and gorilla. And then these are the different variables that we were using. The intermembral index here, this is the ratio of your arm to your leg. It's used as a class of locomotion in primates because classifying locomotion in primates is very difficult. No one can agree on which categories to use, so we used intermembral index instead because it's more of a quantitative measure of locomotion type. Average body mass, age at men-up, which is the age that females reach sexual maturity, group size, interbirth interval, home range size, and day journey length. And then we also had annual temperature, temperature seasonality, temperature diurnal range, annual precipitation, precipitation seasonality, and canopy height. The analysis that we used is a combination of RLQ and fourth corner analysis. So RLQ basically summarizes the joint structure of 3 different matrices. You've got the R matrix, which is site by species. So we used a correspondence analysis on this. And then the L matrix, which is site by environment. This was a principal components analysis. And then the Q matrix is species by traits. So this was a Hill-Smith test. And then the fourth corner basically uses bivariate tests to test for relationships between the environmental traits and the biological traits which occur at each study site. And finally, these were combined. So we used fourth corner. We tested for correlations between the axes that were produced in the RLQ analysis and the different biological traits and environmental variables. And these are the results, firstly, of the RLQ analysis. So you can see this here is the Q matrix. The R matrix, sorry. So this is site by species. So these are where all the different genera fall within this matrix. So here you've got the patas monkey, the baboons, and the mandrills. These are all terrestrial genera. And then along here, you've got the apes and the columbines. These are more arboreal forest-dwelling primates. And then along here, this is the L matrix, so that's site by environment. You've got temperature diurnal range, so the range of temperatures during the day, precipitation seasonality, and temperature seasonality along this side. So these are open, more seasonal environments, so for example, savanna. And then along here, you've got annual precipitation and canopy height. So these are going to be more areas like tropical forest and closed, more stable environments. And then again, on the Q matrix, so this is the species traits, here is terrestrial open substrate and also a folivore omnivore diet. So that's a more generalist diet. These are more towards this side. And then again, here you've got frugivores and folivores. And arboreal somewhere is in here, right up there. So basically, these are all kind of matching up in the way that we would expect. So you've got the arboreal primates towards this side in the forest environments, which are associated with taller canopies and higher annual precipitation. And then you've got the terrestrial, more generalist primates, which are associated with more open seasonal environments, such as savanna, which makes sense. It's what we would expect. When we did fourth corner, we didn't find any significant bivariate associations between traits and environment, which suggests that all of these associations here are not significant, and there doesn't seem to be any link between biological traits and environmental variables. However, when we combined the two approaches and tested the correlations between the axes, what we found is that for the environment axes and traits, there were no significant correlations. However, for the traits axes and environment, there was a significant correlation along the x-axes with temperature seasonality and precipitation seasonality. These were negatively correlated, and annual precipitation and canopy height were positively correlated. So it's suggesting that environment is playing a role in the types of traits that you would get in a given habitat. So if we go back to the beginning of what I was saying about how we think biological traits and environment will interact with each other to determine species survival, what we're seeing is that biological traits seem to be less important, at least at a regional scale, like we've done here, but environment is coming up as being a little bit more important. But there don't seem to be, there's very little evidence here that environmental trait filtering is important for primate communities. And this is surprising, given that it's been shown to be quite important in various other taxa, including birds, bats, and benthic stream communities and insects. So we were a little bit surprised by this result. But when you look at other work, there's been other work that's been done recently on phylogenetic clustering in primate communities. And what we found is while birds and bats have been shown to have a phylogenetic structure, communities of birds and bats have been shown to have a phylogenetic structure that isn't random, so they're either clustered or over-dispersed. Primate communities in general have a random phylogenetic structure, which suggests that they're actually more being influenced by null processes, such as dispersal limitation, as opposed to biological or environmental processes. This could also be an issue of scale dependence, that they might be their biological traits are maybe more important at a local scale rather than at the regional scale. So what we want to do in the future, we're going to first use a GLLM to look more in depth at the relationships that are there, to see if there's anything that's maybe not being picked up in the analysis. The RRQ analysis is really useful for signposting potential relationships, but it doesn't give you a very detailed picture of what's going on in terms of numbers. We also want to look at incorporating traits relating to dispersal ability. There's been a recent paper by Estrada et al., it's a tree article, about the importance of species traits in predicting range shifts. And they've suggested using traits which are important in relation to a species' ability to disperse into and establish in new environments. So looking at things like site fidelity, their reproductive strategy, their level of generalization, and their competitive ability. We also want to look at maybe doing a species level analysis and perhaps bring it down to a more local scale again to see if it is an effect of scale dependence, what we've found. So thank you very much.
Any questions? I was just wondering how you chose which
Yeah, so we did look at, we started off with about 15 different variables. So we went in the WorldClim BioClim database. We started out with all of them, and then we used various correlations to see which variables were sort of correlating with each other, so, and then chose them based on that. So we kind of eliminated them based on, so we found that maximum and minimum temperature was correlating very well with annual temperature, so we knew that there wasn't a big difference.
Sorry, do you want to quickly follow?
Why was it that you used biochem given the coverage for Africa is?
It was just the database that we had available.
On the phylogenetic structure bit, do you think it's because primates are one of the few IUCN groups where they use the phylogenetic species concept rather than more traditional taxonomy? So then it split them into different units compared to other species?
Could be, yeah. There have been, the studies that we've sort of been looking at, there have been studies on birds as well that have looked at phylogenetics, not just at traditional taxonomic methods. It could be that other groups are showing the same thing. It's quite a new concept, so we can't really answer that right now.
Looking at where you had no significant associations.
Yeah, it's
Was it,
there.
any corrections, multiple corrections?
This was before we corrected it. So it was completely, there was no significant correlations at all, which we were a little bit surprised about.
More questions? Do you have any guess for why that happened?
We have a few different ideas. One possibility is because primates, actually, when you look at them, with the exception of a few species, which are the ones that we didn't include, which are the sort of Madagascan primates, which we weren't using in this analysis, they're actually very generalist. When you look at them, they don't have any particular physiological or morphological adaptations. And even the ones that have, like, the ones that were classed as, for example, frugivores, they still eat leaves. When you look at their dietary composition, they have so many different species that they exploit. So it could be that they're very generalist, and therefore they're more resilient to environmental change than we expect, or it's difficult to say.
Anybody else have any more questions? Okay, we'll let you sit down, I think.
All right,
Thank you very
thank
much again. Got about a minute before the next talk's meant to start, so he's going to do a dance.
Did you do your presentation really quickly to interpret that? No.
Yeah, that's what's the point. Yeah. You don't have to excuse to British people who can't dance unless we're like, you know.
Actually, they're recording.
You do have to use the microphone, sorry.
So if you could stand up.
It will pick you up if you move slightly, but don't go all the way to the left.
It only has this room.
I guess it depends if you want people to see the recording. Okay, our next speaker is Emmanuel Fraunhofer. He's going to talk about the eco-evolutionary dynamics of range expansions.
Thank you very much. So as ecologists and evolutionary biologists, we're very much interested in understanding the diversity of species on Earth. But especially a lot of you are also interested in understanding the distribution of these species and their ranges. Now, this is an example from the brown bear, and I took that because I'm going to talk about very uncharismatic organisms later on. And this picture is actually quite misleading for what I'm going to talk about, because it suggests that this range is static. But I guess you all know that if the environment changed, that range might change as well. It might shift, it might expand, it might contract. And this is exactly what we're going to try to understand now. I think a perfect example for such a dynamic range comes from Australia and the cane toads that were introduced on the very eastern side of the continent from their natal range in Central and Southern America, and they subsequently spread all across that continent, destroying local communities. So that's an interesting study object for ecologists, but it's also an interesting study object for evolutionary biologists, because those cane toads actually showed evolutionary changes during their range expansion. I think that in order to understand species ranges, it's very helpful to take a metapopulation perspective. So to imagine the world as being spatially structured and set up of individual patches of habitat that can be connected via dispersal. And I think at the end of the previous talk, we already heard that dispersal might be something that is important for species ranges. And so if we do that, I think two things become quite immediately clear. That colonizations are an important driver of ranges because they lead to range expansions. And on the other side, extinctions are important drivers of ranges because they lead to patch extinctions and range contractions. And while this might seem trivial to some of you, it also highlights that dispersal is really important because dispersal is really the driver of colonizations, and dispersal may also lead to local patch extinctions. So a few years back with some friends and colleagues, we took this idea and said, well, it's a huge chance of merging what range biology, basically, and everything that we know about dispersal ecology and evolution, because researchers have been studying dispersal in both contexts for quite a while now. And if we put all of that in that framework, we come up with three broad categories. So abiotic factors and intra- and interspecific biotic factors that can impact dispersal and thereby ranges. And those are just some of the most important boxes, and you don't need to read all of them. Just on the left side, you find what has basically been studied in metapopulation ecology, so patch area, patch isolation. In the center, some aspects that have been studied by population ecologists, such as density regulation. And on the very right side, factors such as species interactions that are more at home in community ecology. You also see that there are black arrows, and those are ecological influences, and red arrows, and those are evolutionary influences. And so, quite clearly, if those two processes happen at similar timescales, we will have to deal with interactions of ecology and evolution, and maybe even feedbacks between ecology and evolution. So today, I'd like to focus on the left side and integrate one box from the center, which is information use, and I will explain to you why in a second. A lot of models, a lot of very simplified theoretical models on range expansions assume homogeneous habitat, and some of the experiments that have been done as well. But you all know that this is pretty much wrong, that there are gradients everywhere, gradients, for instance, in altitude, in temperature, in humidity, or maybe in fragmentation. And so if we want to understand ranges, we need to include gradients. And this is what a master student of mine, Nikolai, wanted to do. So you imagine, for simplicity, a linear landscape. Those are patches connected via dispersal. And now we want to introduce a gradient, and a gradient that has an impact on fitness-relevant aspects, and we'll just take mortality for simplicity. And while this might work, say for temperature, at the very same time that this gradient has a fitness-relevant impact, it might also transport information. Imagine a butterfly flying in a temperature gradient. It will most likely not fly into areas where it is really maladapted to. It might turn around and choose to settle somewhere else. And this is the idea that we're following here. So this is an informed scenario, as we call it, and in nature these two things are, I think, always coupled. However, in models and maybe in some experimentation, we can try to uncouple them and to understand the relative importance of this direct effect of the gradient and this indirect effect via information and the use of information. And we will contrast that with a third scenario, which is a control that has no gradient and therefore also no information. So we first started out by doing some modeling. And so we assume a linear landscape that is empty, a population starts here and can spread throughout that landscape. We assume that dispersal is a trait that can evolve. We assume that there's a trade-off between dispersal and foraging. So you can either disperse a lot or forage a lot. Dispersal is costly in that way. And in the informed scenario, we assume that dispersal is completely plastic. So individuals know everything about densities around them and the gradient and can just take an optimal decision of where to disperse to. I'm going to show you some results of those simulations, plotting here population density over space, and here the evolved dispersal rate over space for the three scenarios: control on the top, the uninformed in the middle, and the informed one at the bottom. So the first thing to realize is, of course, we won't see anything here because here we assume plasticity, so dispersal is not evolving. If you look at what happens, then quite quickly we see two patterns emerging. Let's start here. So dispersal seems to go up from the range core to the range margin, and this has been seen in the cane toads, for instance, and has been called spatial selection. So imagine there's variation in the dispersal trait. Just because the fast ones are really fast, they will outrun the slow ones and be at the range front, and they will mate with each other. And because competition is very low, remember the landscape is not inhabited by anyone else, they have a huge fitness advantage, and that might lead to kind of a runaway selection process for increased dispersal. And then we see this unimodal pattern here, this high density at the range front, and this is due to that trade-off between dispersiveness and foraging ability. Simply, the individuals back here, they depress the amount of resources so much that their equilibrium density is low. And as the range expansion proceeds, we start to see an effect of the gradient that kicks in pretty late, and we immediately see an effect of the information. This information use inverts the distribution of densities in comparison to the control, and it also inverts the distribution quite late into the expansion of the gradient scenario. So this is, if you want, a model, and some of you might say this is just another model, and we might wonder, well, is there evidence? And I'm not just talking about correlational empirical evidence. I would like to have experimental evidence for that. So that's quite challenging. Some of you are nodding. It would be nice to have that, right? And so here we use a trick. We don't go out in the field and study elephants, but we take those large spatiotemporal scales and collapse them into the lab and use microscopic model organisms, and in this case, Tetrahymena freshwater ciliate, and built microcosm landscapes that they can invade. So reproducing what happens outside at very large scales, potentially at very small scales in the lab. We use video analysis to collect data, so these are tracks of individual protists in those microcosms. We collect behavioral data, morphological data, and obviously population dynamic data in that way. So before you tell me that, you know, this is nice, but they will certainly not be able to use information for our third scenario, they actually do. So the information we will take is the presence of dead conspecifics. So we did some preliminary tests here, had a two-patch setup with a control and a treatment where they had dead individuals, and they could decide to go either to the control or to the dead individuals. And if the dead individuals are a cue for mortality, they should avoid those. And from a certain concentration onwards, they chose the control. So we seem to be able to use dead individuals, or the scent of death, to signal that there's a mortality effect. I'm going to show you exactly the same kind of data, just now experimental data. So this is medians over 6 replicated range expansions from the control scenario, a gradient scenario where we just apply increasing mortalities and do not put the dead individuals back in, and a scenario with information where we just put the dead individuals back into those test tubes. Let's see what happens. Again, we see this pattern of increasing dispersal towards the range front in all 3 of those. We see this unimodal distribution here. And if we go forwards in the range expansion, the increase in dispersal seems to be very clear, the unimodal pattern as well. And here again, they seem to stay in the range core and not expand very much. So that fits pretty well with our expectations. We also find the same very small difference between the control and the gradient only, so that the gradient has a rather limited effect on the distribution of densities throughout that range. So some of you might be wondering whether this is really evolution or just plasticity, and so we conducted a common garden experiment after the end of the evolution phase and re-assayed individuals And the difference that we find thereafter should be something that is related to genetic differences and not plasticity. And so that's what we did right here. And you see in the two scenarios where we'd assume evolution to be going on, we see a significant difference between range core and range front populations, blue for the core, range for the front in dispersal ability. And we don't see any difference in the scenario where we assume that everything is plastic. So that seems to make a lot of sense in that context. There was some plasticity going on here because they seem to increase their dispersal ability in the beginning, but then this line was pretty flat when they realized that they shouldn't disperse more into that gradient. So to summarize, in the control, what we find is an eco-evolutionary feedback loop. So this ecological process of the range expansion selects for increased dispersal at the range front. This feeds back on components of the functional response. Remember, there's this trade-off between dispersal and foraging ability. And then it feeds back on the ecological pattern that we see, and it inverts the distribution of densities. This might seem like a detail to some of you, but if you think about the establishment of stable range borders due to migration load, so the invasion of maladapted individuals from the core, then if the distribution of densities is inverted, this will play out very differently. If we include this part about information and the gradients, this basically kills the evolution side and kills the eco-evolutionary feedback loop and makes the whole distribution stay in the range core. With this, I'd like to thank everyone who is involved, especially Nikolai, who did a lot of the experiments, and all of the Ultimate group, and you for your attention.
Got time for a couple of questions? Anybody?
How the hell do you identify the individuals in those microcosms and take them out or take them back and put them back in?
So basically, this is done via pipetting. We have you apply a gradient, you say you kill 10%, 20%, 30%, and take individuals out via pipetting, replace that volume with new medium, and in the other case, we use sonication to kill the individuals, try to not change anything about the temperature so that it changes maybe secondary compounds or metabolites or whatever we have in there, and then put that back. Well, the microcosms are not that small, so we don't have to work under the microscope for that. It's just the data collection happens on the microscope.
Questions?
Yeah.
You started one of your examples with the totems in Australia, with expansion over time, being an invasive species. Have you tried to apply your system in the way, basically the results of the system on the expansive rings of its, like the expanding range of that totem in Australia? I think I even know because they have kept track of like where does it occur that year, next year, following year.
Yeah, so qualitatively, it fits from the evolution of the dispersal ability. I don't know exactly what you mean by applying it. So taking that and parameterizing a model or something like that.
Yeah, because you started off with species range sizes and then
Yeah.
down very small scale.
So
So what's the feedback loop back to the macro scale?
I think this is a proof of concept that these, for instance, this eco-evolutionary feedback loop has an impact on these large-scale patterns, and it is also a hint for people working in the field and working with real species, if you want, that if some models do not fit, there might be a level of complexity related to dispersal and maybe even a more complex dispersal behavior that would need to be included. I wouldn't use that to parameterize a model because the evolutionary trajectories will depend very much on the genetic architecture, on mutation rates, and that we don't know, but it might be species specific. It's very certainly different if you evolve cilia or if you evolve longer hind legs, right? There will be different constraints, but I think like as a proof of principle, it works out pretty well.
Okay, well, thank you very much for that. Macroecology compatibilata. I should have practiced that beforehand. Very good. I didn't get that right the first time I worked with this species. Right. So every species is adapted to very specific environmental conditions. So, for example, it occupies a range that includes its favourite food, its preferred climate conditions, and together that makes up the species' geographical range. Now, when the environment changes, the optimum location for the species to be in is expected to change as well. And when that happens, a species has a couple of options. So it can try and migrate along with these shifting environmental gradients. If not, it can try and it can stay in the same place but try to adapt to the new conditions. But if migration or adaptation are not good options for the species, it might end up in a location that is suboptimal. And in the worst case scenario, this might actually cause the species to get extinct. Now the problem is we don't have a lot of empirical data on these processes, especially if we go over timescales that are longer than a couple of 100 years. And the reason for that is that we basically need really high resolution fossil records from loads of different locations. And for most species, this is simply not feasible. So in my PhD project, I study planktonic foraminifera. These are a group of calcifying zooplankton. They're about the single-celled organisms, but they're about the size of a grain of sand, which is actually quite big for a single-cell organism. Like this one over here is about one of my favorite examples. It's about 0.5 millimeter across. And this single-celled thing is eating a little shrimp. So they build these calcium carbonate shells, and when the individuals die, the cell sort of, well, disintegrates, and then what you're left with is these sort of fossil shells. Now they sink down to the bottom of the ocean floor and they accumulate in the sediment to make these huge layers of fossilized forams, and they can stretch over millions of years back. And then as an additional advantage, because they go so far back, we can sample them continuously over past climate events. So we get really long-term records of how species responded to climate change. The species I'd like to focus on today is Globicanella ponticulata. Its range was about 4.7 to 2.4 million years ago. And especially the timing of its extinction is quite interesting because that occurred around here, right around the intensification of Northern Hemisphere glaciation, so when Greenland and North America became glaciated. So early Pliocene around here, temperatures were a couple of degrees warmer than today. We didn't have a lot of glacial interglacial variability. That sort of started to change around 2.5 million years ago. So this one here, glacial event G6, that was basically the first large glacial, and then after that, glacials and interglacials sort of seem to oscillate on like about a 40,000 year cycle. And then shortly after that, we get the first 3 major glacial phases, and that's when this species goes extinct. So we wanted to study what happened to this species before it became extinct. So we studied an interval of about 500,000 years, and that includes this entire transition from like warmer temperatures to the onset of glacial cycles to big glacial cycles. So here there's nothing, not much going on yet. Then we've got the onset of the glacial cycles. Then here we've got 3 really big glacial cycles, and then the species goes extinct. So what we wanted to do was to see how the geographical range of the species shifted through these climate transitions, and also how well it was doing within its range. So to do this, we looked at 2 indicators. So we studied abundance and size. So they're both indicators for how well the species is doing. Abundance, for example, basically the more individuals that are, the happier the species is. And then basically the same goes with size. So there was this early study by Hecht in 1976 that showed that foraminifera sort of reach an optimum size at a certain location. For example, this species seems to be largest around the mid-latitudes. And then later on, a study by Denny Schmidt and colleagues showed that maximum abundance and maximum size seem to occur at the same place. So together, abundance and size are quite good indicators of how well the species are doing. So in this study, we looked at 6 different sites throughout the Atlantic, all different latitudes, different oceanographic settings. Depending on the site, we used a sample resolution of about 5 to 10,000 years. Then, to account for all the variation that we found in the species, we used about 50 to 100 specimens per sample. And that gave us a nice working total of 16,082 individuals. And then we looked at abundance and size for all of them. Right, so starting with the first results. So early on in the study time interval, not much glacial activity yet. So different colours represent different sites. And we see that there's quite a clear peak in abundance and maximum size. It goes around these two, so red and green. It corresponds to those 2 sites here in the mid-latitude of the North Atlantic. Then there's a couple of sites higher and lower latitudes, so black and blue around here. We're still doing all right. But the abundance is a bit lower and sizes are a bit smaller as well. And then when we go even further down to the equator, the species was really starting to struggle. They were really difficult to find. So these 4 samples here were the only samples I was able to find the species. And they were really small. They looked quite sad, actually. So here there was still quite a lot of variation between sites. Then further on in the interval, so when we get the first glacial interglacial cycles, we see that that optimum, what used to be an optimum in the mid-latitudes, has now disappeared. So they're all sort of merging towards the same sort of size. And then the equator site is still not doing very well. And then going towards the final stage, before the species goes extinct, with those really big glacials, I see the same pattern, but you also see that the variation in size is sort of starting to narrow. So it means a loss of morphological variation and basically doesn't really mean well for the species. So it goes extinct not long after that, and the different populations became extinct at different times. So again, time is going left, it's plotted in the geological way. So the first population to disappear is the black population, so site 981 at the high latitudes. So that's the first one to go. That's followed by one of the mid-latitude sites and the equator site. So the equator population is already doing so badly that we're bound to go at some point. But it does mean that now the northern hemisphere and the southern hemisphere populations are sort of separated. Then the other mid-latitude site goes, shortly followed by the southern hemisphere mid-latitude site. And then the only population that's left is this one, it's site 659. And that persists for another 50,000 years or so. So it seems that briefly the optimum of the species has shifted from sort of mid-latitudes to the subtropics. But apparently it wasn't perfect because in the end the species did go extinct. So these results seem to imply that climate change had a big impact on the species and that it caused some quite big range shifts. The thing was, with the intensification of northern hemisphere glaciation, it was basically a major reorganization of the global climate system. So obviously, global temperatures dropped, but nutrient availability in the oceans started to change as well. Productivity became different. Atmospheric CO2 levels started to change, so that affected the acidity of the ocean, and therefore could have impacted the calcification mechanisms of the forearms. So we'd quite like to know which of these parameters had the biggest impact on the species, or if it was maybe a combination of everything. So to answer that question, I'd like to focus on these 2 sites, so mid-latitudes and high latitudes. And the reason for that is that for these sites, climatic reconstructions already exist for the interval that we studied. So there are site-specific reconstructions of temperature and also of productivity. And then for the same interval, there's also a reconstruction of global atmospheric CO2. So we can use these 3 to sort of study how the species responded to these local environmental changes. Right, so starting with the northern hemisphere, high latitude site. These are abundance and size from this study. So we see that size is quite stable, abundance is actually quite variable. There's a couple of times where the species just completely disappears from this site. Then we've got the existing reconstructions of atmospheric CO2 from Martinez Botti and others. And then we have sea surface temperature and productivity that are specifically reconstructed from this site. So to figure out how much of the variance in either abundance or size was explained by climate, we used generalized linear models. So you see there's quite some differences in response between size and abundance. At this site, size doesn't really care. So even when we look at the combinations of all these parameters and their interactions, like less than 5% of the variance is explained. So it doesn't really seem to be responding to climate. But then when we look at abundance, all the, like the total model and their interactions explain more than 20% of the variance, which is actually quite a lot. So in this case, at least abundance seemed to be responding quite strongly to climate. Then going to the mid-latitude site. So this site is what the previous species optimum was. So we expect them to be a bit more in the comfort zone here. So we'd like to know if the responses here were any different than at the high latitude site. So again, abundance and size from this study. Here we see that abundance is really, really stable until the very last sample where we can find them. It just drops off and they disappear. But size does something funny here. So they start out as quite big. And then suddenly there's this sort of drop towards like smaller shell sizes, and then they stay a bit smaller and then disappear. And then again, CO2, productivity, and sea surface temperature. Now when we look at the variance explained, we see quite different patterns. So abundance and size seem to be responding, seem both to be responding. Actually, size seems to be responding a bit more strongly. And then when we compare that between sites, so there's a higher response in abundance at the high latitude site and a higher response in size at the mid latitude site. So to conclude, it seems that climate change due to the intensification of northern hemisphere glaciation had a big impact on the species, and that it was a combination of loads of different environmental parameters that caused the range shifts before the species became extinct. Thank you. In particular seems to be very sensitive to climate change. We did get another species over the same time interval that did survive through all these glacial interglacial variations, and they didn't care. They just went through and nothing changed. They were absolutely fine. Right, let's back out of it again. Very strict instructions to keep to time, so following the clock to the very second.
We could have a countdown from the audience.
Right, good. Alright, so yeah, we need to stand up my phone so you can be recorded. So our next speaker then is David Wilkinson, talking about visiting a Darwinian obsession, waterbirds and plant dispersal.
Okay, so this is a talk with three authors. I'm presenting it. One of the other authors, Andy Green, is just sat at the front here, but as far as I'm aware, Adam is somewhere in Hungary and certainly not at the meeting. So what I want to do is to talk about some recent data that we've been collecting, and everything that you'll see in this talk is actually unpublished, on water birds, and particularly in the context of seed dispersal. So this is a biology talk. So classically, you've got two starting points. I'm not doing Aristotle, so I'll start with Darwin. And Darwin, as you know, had this thing about islands. And if you're interested in islands and biogeography of islands and evolution on islands, how things get there is important. So Darwin had a big interest in dispersal, and he spent a lot of time doing a lot of really quite innovative small-scale experimental studies, both in the grounds of his house and the surrounding countryside, and also at London Zoo, looking at seed dispersal and the way in which birds potentially move seeds around. Because Darwin went, well, hang on, plants get to islands, plants don't move, plants don't swim, birds are probably quite important in moving things around. And so a lot of early work on seed dispersal was done by Darwin. And indeed, within this country at least, there's not been a lot of stuff done since, with the exception of stuff on things that are obviously moved by birds. So things that move with berries, that birds obviously eat and move around, they've been studied extensively, and particularly sort of classic work by David and Barbara Snow some decades ago. But other than that, there's not been a lot of work done in this country. So, classically, the sort of textbook approach is that if you're a bird or any other animal, you can move things around in one of two ways. You can move it around externally, and you can see, at least the people sat at the front, can see we've got duckweed all over the bill here and on its feathers, and that moves to another, the duck moves to another pond, and we have duckweed again in that pond. So we can have external transmission, and clearly also it can eat stuff. And so the duck can eat seeds, the seeds can go all the way through the duck, out the other end, and potentially colonise the site. And all sorts of birds can do this, but within this talk I'm focusing on water birds. So what we've done on the data I'm going to show you is a whole collection of bird droppings collected and analysed, a fairly simple approach. So collected from the field, brought back to the lab, sieved, and then under a low-power microscope, the seeds picked out and identified. Clue, Adam is from Hungary. In Hungary, they still teach proper old-fashioned botany, so we didn't have to PCR the seeds to identify them. We had someone who could do it the old-fashioned way. And then we would germinate the seeds on a non-nutrient agar to see whether we can get them to grow, because clearly we're interested in whether they can actually grow or not. Those of you wise to the ways of the world will say, well, this is a paper with 3 authors, 2 of whom are professors, 1 of whom is a PhD student. Guess who sieved all the duck poo? Okay? And then put in an awful lot of work. And so the data underlying this talk is nearly 700 bird droppings, mainly from Britain, from around other places as well. So I mean, if in the bar tonight, conversation flags, you feel the need for sort of say something, yeah, and you want to say, well, I attended a really shit talk this afternoon. This is the one you can use, because we have loads and loads of bird droppings here from a wide range of species, including Canada geese. Oh, this is my fallback in the PowerPoint dies, including lapwings and big-footed geese, whooper swans, and a whole range of things. And we've got seeds, which are inverted commas, in case there is a proper botanist here who will point out that many of these things are fruit, from over 60 plant species. And so let me give you some summaries. I'm not going to get bogged down in the details of the data here, because I'm really more interested, because it's me, in the ideas that start to underline them. But we had over 1,000 seeds, and of those, over 15% germinated. And that's clearly a minimum value. That's just what we could get to grow on non-nutrient agar. Most of these seeds are not from wetland plant species. So you might naively assume that water birds are going to move stuff around that are wetland plant species. The majority of our seeds are not from wetland taxa. And for botanists here, and, oh, there we go, familiar with Ellenberg's values, Ellenberg's F, which is a measure of how wet things like it, out of our 50 taxa here, then the majority of them come from F3 to 8, and those are all drier than F9. F9 is what's described from wetland there. So the majority of our seeds are coming from taxa that are not wetland species. So I'll briefly show you sort of 2 little highlights that make little nice stories before getting to the bits that actually sort of matter from a theoretical point of view. So most of this is on seeds or seeds and fruit, but we have some other data as well. This is the southern end of Lake Windermere in the English Lake District at Felfoot. And from mallard droppings there, we extracted little bits of bright green and probably live-looking moss. And indeed, in one case, when we scattered these over non-neutral agar, we managed to actually grow moss. So this is moss that has been eaten by mallards, gone all the way through a mallard, come out the other end, and is still viable and will still grow. Now, particularly for younger people in here might think, well, actually, the problem with doing science in the early 21st century is everyone has done more or less all of it. Absolute rubbish. There is no record in the scientific literature at all that we can find, unless if it is somewhere very, very, very obscure, of someone growing moss from bird food that's been all the way from birds. It's been done for bats, rather bizarrely, but not for birds. And so this is science that requires nothing other than bird food, sieve, no power microscope, a bit of jelly in a petri dish, and to show something completely new, that moss going all the way through a bird is still able to grow. And clearly, given the amount of moss that moves around within birds, lots and lots of fragments, we have many other samples with live-looking moss in, it's just that this is the one we managed to grow, it may be an additional way that moss makes its way around the world. There's also clearly quite a big interest in invasive species and non-native species. So up the top here we have buttonweed on the Wirral here, and the field at the top there is a roosting site for black-tailed godwits. You can see there is the distribution of butterweed across Britain from the SBI's New Atlas of 2002. And there's the Wirral. It's one of the centres of it in this country. And lots of the droppings of godwits contain the seeds of buttonweed, and put it on your agar. You get cute little baby buttonweed seeds coming up on your, and they grow easily. We've got loads and loads of those growing. And so we sort of suspect that waders and other things are a major way of moving this thing around the country. So what I want to do is to now make a number of points that sort of matter. So I've already made this point, but if you think about water birds, you think, well, all right, water birds are important for moving stuff. They're important for moving stuff that are wetland plants. But most of our species of plants are not wetland species. It's more important than that. If you look in the big databases of plant ecology, this is a French database, you get qualitatively the same result, though playing with multiple other databases. Of our taxa, 54% of the taxa that we have out of the bird droppings are things that are not animal dispersed. Basically, the books are wrong. Now that worries me, it worries one or two other people with grey hair in here. If you're not worried, I'll rephrase it. The PDFs are wrong. The websites are wrong. The assumption that is made in distribution of plants is that you look at the seed and you see this has a wing, it's wind dispersed. This has a fruit, it's animal dispersed on the inside. This has a little hook. You can get it on your clothes or your trousers or your dog when you go out for a walk. It's dispersed on the outside of animals. But in fact, most of our seeds, most of our species are from things that don't have adaptations for seed dispersed, are not called animal dispersed within the databases. And it's actually more than that, because if you look at the things that are in that database that we've got as animal dispersed, most of them are only there recorded as dispersed on the outside of animals. They have adaptations that people assume are therefore the outside of animals. And again, if you look at these databases, loads and loads and loads of species are recorded as no obvious means of dispersal. They haven't got a sail, they haven't got a hook, they haven't got a fleshy fruit. But that doesn't stop them being dispersed by animals. But all the modelling, all the attempts to predict things about changes in plant species, ranges with changes in things like climate, if they're using data on seed dispersal, use these sorts of databases. And yet the databases they're using are completely and utterly inadequate, because they say this is dispersed by wind, this has no obvious means of dispersing, the pollen doesn't go over. All of these things, or at least a very high percentage of them, are dispersed by birds, and therefore can move really, really long distances. And this is sort of a bit worrying, at least a bit worrying for me, because I am, at least sort of one and a half theoretician. And it means that if you want to model these things, I'm afraid there is actually no other option than empirical data. There is no other option than old-fashioned natural history and a PhD student and a pile of bird poop and a sieve. Or if you can't identify the seeds, PCR, if you want to be high-tech. But we need empirical data if we are going to be able to actually say which species are. There may be some that for various reasons of their biology and natural history aren't easily dispersed by birds. But the assumptions we're making about dispersal at the moment just don't hold up. And if you use these databases, which are based on these morphological descriptions of the way things move, it's not telling you the right story. And so given, and these are some of the pink-footed geese at Martiniere on the sites we use, there are enormous numbers of them, and they go all over the place, and they take seeds with them. Given the enormous numbers of birds flying around places, and focus on water birds here, but in other areas I have interest in other birds moving things around, and if you want to move across the equator, I've tried to do it in a supercomputer with wind flow for little airborne things, and it's very difficult. But colossal numbers of birds migrate across the equator and down into Africa from Northern Europe and things every year. So we are greatly underestimating the importance of birds in moving seeds and other organisms around. And in hunting bird food, you'd think bird food is easy, but in hunting bird food alone, we've had an enormous amount of help. At least some of you have managed to get onto this thank you slide. And they do seem to drop when they're doing long-distance flights, which is quite interesting.
So they're probably as good long-distance dispersers as large mammals, but how do you explain the hundreds of kilometres that not have to do between islands?
But actually, and my goal over here is that man is being involved with that, so go on.
You might want to look at a tree paper by Viana Tower, came out a couple of months ago. The models predict maximum dispersal distances over 1,000 kilometres for many seed types. Because these distributions have a very long tail, but the actual time it takes for a seed to come out is actually with a very long tail. Proctor in the 60s actually found seeds that took over a week to come out from shorebirds.
And so it could be lots, because let me disagree with my self of 20-odd years ago, where the last time I played with these sorts of things, I was arguing for birds being really, really important in moving lots of things with morphological adaptation, things like wind dispersal. But at that point I said, well, it's just odd, rare events which are very important to biogeography and range size. But the sorts of numbers of wind dispersed or other dispersal mechanisms that aren't supposed to be anything to do with animals that we're getting in these birds, and the colossal numbers of birds you've got flying around the place, I suspect I was overly cautious in saying, well, it's just rare events. It's actually probably really rather common. Yes, but doesn't feature in anyone's attempt to use a database to model these things.
I was just wondering, since we use agar plates to grow the seeds, do you know if there might be any bias against?
Yes, there will be. So there are clearly two approaches. One is to just use one method, then you're comparable across, or they would take seeds and try various different things. In this particular approach, we just use non-nutrient agar because previous experiments by Andy and Adam had shown that that actually worked better than, say, damp filter paper or whatever. So it's one method that works quite well. So as I said, the germination data is a minimum. It's just what happens on non-nutrient agar.
I don't think we've got time for any more questions, but thank you very much to David.
Yeah, I just want to check this out. So this is the okay.
Yes, it's forward and backward. Point it and that'll turn it later.
Okay. Thanks.
Okay, and our next speaker is Neil McGuinness. He's going to talk about dispersal versus environmental controls of Scleractinian coral species assembled into composition.
Thank you. So the background to this work is the fate of corals under climate change and the coral reef ecosystem. You're probably all aware of the widespread recent bleaching of corals that's been going on. This is actually the third global bleaching event, with the first one happening in 1998 and the second one in 2010. So coral bleaching occurs when corals get stressed, and in response to the stress they expel symbiotic algae from their tissues, and unless the stress recedes and they reacquire symbionts, the corals die. So under these past two bleaching events, there's been widespread death of corals and reduction in coral cover and significant effects on the ecosystems. This latest event is longer lasting and more severe than the previous two bleaching events, and as we continue into the future, bleaching events are predicted to occur more frequently with greater severity, and climate change is believed to going to occur at a rate that exceeds corals' ability to adapt. So that means that corals have been, it's been suggested that corals will have to shift their ranges in order to survive in the future. So I wanted to investigate what evidence there is for environmental control of coral distributions today, and if there's evidence for dispersal limitation in their distributions, because that would have consequences for their ability to disperse to these climate refugia. So I focus on assemblages rather than species richness, because that enables us to distinguish between areas of similar species richness but different composition. Previous work that's taken this approach has identified the broad faunal provinces in the Indo-Pacific, which you can see in this image here, with coloured regions representing distinct faunal provinces. When the authors investigate what factors might generate these distributional patterns, they found strong support for geological features. So clusters based on geological features such as volcanic island chains correspond pretty well to the breaks in the faunal provinces that you can see in the top image there. But when environment was investigated, there was very limited support for its role in generating biogeographic patterns in corals, which you can see in this lower image where the colours representing distinct environmental conditions don't correspond at all well to the faunal provinces. So I found this pretty surprising. We know that extremes of temperature and salinity are fatal to corals, and high concentrations of nitrate compounds in the water can lead to macroalgal overgrowth. So it did seem surprising that there was a small effect of environment here. But it could be that it depends on the scale of analysis, the effect of environment. So perhaps at smaller spatial scales, environment is more important than we're seeing here. Another factor that was investigated was distance between habitat. And again, this was found to have only relatively weak support for generating these biogeographic patterns in corals. So this distance between habitat is a commonly used proxy for dispersal, with the idea being that as distances between habitat increases, then dispersal between those locations will decrease. So this could mean that dispersal isn't very important in generating biogeographical patterns in corals, or it could mean that the proxy is not great. But unfortunately, this has been one of the most commonly used proxies, I would say, in investigating dispersal, but that's because dispersal is really hard to measure. So I'm studying Scleractinian corals, as you've heard. Here you can see broadcast spawning coral, which probably make up about 90% of all species in the Indo-Pacific. So they release their egg and sperm into the water, which float to the surface, and fertilization takes place. And planular larvae develop. So these microscopic larvae are then dispersed by tides and currents, and the direct tracking of this is not feasible, basically. So we've had to use these other proxies. But recently they've been developed in modeling dispersal. So in 2014, Sally Wood et al. Published the first global model of coral connectivity. And this is a biophysical model, so it takes into account data on larval longevity and acquisition of competence, and also oceanographic data. So what they do in this model is they release larvae from every known reef location in the world over an 8-year period on a monthly basis, and then track the corals through the water and record their destination. So that's what you can see in this image here, where the reds indicate high concentration of coral larvae. So this gives us an understanding of the connectivity of reef locations, and it enables us to test hypotheses of dispersal in a manner which hasn't been possible previously. And we've been lucky enough to work with Sally Wood and have access to this data. And why might this be important in biogeography? Well, barriers to dispersal could limit range expansions and isolate populations. And successful dispersal connects populations, supporting their persistence, and allows for range expansions. So I had the following research questions. Is the connectivity of reefs correlated to the similarity in assemblage composition? Are similar environmental conditions between reefs correlated to similarity in assemblage composition? Which factor correlates most highly? And does scale have an influence? So in order to do my analysis, I generated reef clusters, and this was necessary because of differences in the resolution of the data for species ranges, which is fairly low resolution data, compared to the connectivity data from Sally Wood et al., which was a much higher resolution. So in this figure here, you see 4 reefs exchanging larvae. I've clustered reefs based on where they receive larvae from, under the premise that reefs that receive larvae from similar locations will have similar assemblages. So we can present that in a connectivity matrix, and then using an appropriate similarity metric, we can compare every reef and understand how similar they are in terms of where they receive larvae from. Using that, generating a dendrogram and extracting clusters. So we can use this approach to generate different numbers of clusters, and that allows us to sensitivity test the analysis, and that's how I also investigate the effect of scale here, by using different numbers of clusters. So at the very broad scale, I generate these clusters for the Indo-Pacific, where reefs within those coloured polygons are more similar to each other than they are to different coloured polygons. And I do this for 6 different clustering sets. So I use these clusters for the remainder of my analysis. I'm going to have to accelerate. So having generated those clusters, I determine their connectivity using the Wood et al. Data, and generate network models of connectivity, where the nodes represent the clusters from above, and the arrows indicate the exchange of larvae between clusters. So I do that for all of those cluster sets. But you can draw some really useful metrics out of the network analysis. So if I'm interested in the connectivity between Hawaii and French Polynesia, we can track dispersal through the network, even though they're not directly connected, and extract the shortest path, taking into account connectivity between all of the clusters. So we do that for all clusters. I overlay the clusters with environmental data and determine the average within the clusters, and then, similar to before, compare all of the clusters in a pairwise manner. And in a very similar approach, I overlay, these are the species ranges of all Scleractinian corals in the Indo-Pacific. So I overlay those onto the clusters, sum them, and analyze the similarity of the clusters in terms of their assemblage composition. And just before I can get into the final analysis, I have to take into account the potential for spatial autocorrelation of the data. So I measure the shortest distance through the water between the centroids of all of the clusters, which I then control for later. So I conduct my analysis using mantle tests, or partial mantle tests. Mantle tests find the correlation between matrices, whereas partial mantle tests allow for the control of a third matrix, which in this case is the distance between those clusters. I'll skip through that. So when we look at the results for the dispersal model, I find an R squared M of between 0.42 and 0.7, with a mean over the cluster sets of 0.53, and it was significant for all cluster sets. For the environmental model, I get an R squared M between 0.09 and 0.37, with a mean of 0.29, which is significant for 4 of the 6 cluster sets. When we plot this, you can see that at the scales that I analyze the data, connectivity is more important than environment at all scales. However, you can see that scale is really important where environment becomes much more important in determining the assemblage composition of corals as the scale gets smaller, and connectivity becomes less important as the size of the cluster decreases. So coming to my conclusions, connectivity between sites is strongly correlated with the similarity of assemblage composition. Environmental factors also correlate with the similarity of assemblage. Connectivity correlates more highly at these broad scales, but the relative importance of these factors does depend on the scale of analyses. So I think there's some broader implications of this. It's interesting to think whether this might generalize to other organisms and systems. There are many marine taxa that exhibit similar biogeographical patterns to corals, and a number of these have similar life history traits, such as sedentary adult phases and dispersal juveniles. So it could well be that these organisms also are dispersal limited. And does this generalize to terrestrial systems where passive dispersal could take place? And it could be relevant also to biogeographical theory, such as island biogeography, the position of islands relative to prevailing ocean currents and winds could have significant impacts on the immigration rate. And there are practical considerations. This certainly indicates that corals are dispersal limited, so conservation planners need to take that into consideration in the placement of marine protected areas to allow for stepwise dispersal of corals to climate refugia. And with that, I'd just like to say thank you very much for listening. Thanks to my supervisors and also to our collaborator, Sally Wood.
We've got time for one quick question for Neil. Questions? Yep.
Neil, are your conclusions consistent with other studies using genetic data?
I don't know, to be honest. Sorry. I'm not aware that genetic data is available on such a broad scale for type of dispersal. So the connectivity studies that involve genetic data are typically at a smaller scale than this, just because it's so difficult to generate a suitable amount of data when you're looking at patterns across the entire system.
Thanks, Neil. Okay, our final talk of this session then is Sally Keith, and she's going to talk about global cues for coral mass spawning.
Thank you. Okay, hi everybody. Thanks for staying until almost beer o'clock. All right, so in macroecology, we're really good at this. We're looking a lot at where species are. So we just saw Neil put up this map, which is an example of where coral species are. But more and more we're getting into realizing, and a lot of people are actually looking at this now, we need to also be thinking about what is it that species are doing that enable them to be in those places. And so we really need to sort of get more into looking at process and thinking about what's really going on. And so I do that mostly using coral reefs, but it applies to a bunch of different organisms. So the first thing they need to do is they need to reproduce. So they need to actually produce the next generation, otherwise they're never going to exist in that place. They need to also disperse, so we just heard a bit about that, and they need to survive. And this all needs to happen. So we need to look at all of these aspects of the life cycle to be able to really understand macroecological patterns. So as I said, you just heard from Neil about dispersal of corals. I'm doing a bunch of work looking at coexistence of coral and reef fish. But what I'm going to talk today about a little bit more is reproduction and potentially think about how that can lead to diversity distributions and abundance. So if we're doing that, we really need to think about these early life stages. And so dispersal and reproduction are happening really early on. And I think maybe we've got a bit of a problem because most macroecological data we use is based on adult occurrence. So anything we're downloading from GBIF, when we're using these kind of large-scale data sources, we need to be aware of this fact, I think, and maybe think about it a little bit and think about whether that's important for our given taxa that we're looking at. So, yeah, is it possible to really infer underlying processes from these data? Can we say anything about dispersal or reproduction if we're looking at adult occurrence data? And this is particularly important for sessile species, which are often the habitat formers. So corals and plants don't move around, right, once they're there. So the stuff that happens that determines where they are, a lot of it happens early on. So we need to think about things like corals and their larvae and that kind of stuff. Right, so coral reproduction. So we have a couple of different modes. We have brooded larvae. So this is where we have internal fertilization. And they just sort of fall out of the adults and don't really go very far. And then we have broadcast spawning species where we have egg and sperm packets released into the water column and external fertilization. And these guys actually have mass spawning events. And as you heard, they actually constitute about 90 to 95% of all the coral species in the Indo-Pacific, and they are the main habitat formers. And so it's really important to try and understand these mass spawning events. So what are they? So they're defined as the synchronous release of gametes by many species of coral in one evening between dusk and midnight. It's a pretty amazing spectacle, which I'm sure you've all seen on some David Attenborough program at some point. But we really know a surprisingly little amount about how this works. In the morning, it kind of looks like this. So this isn't what you see on the David Attenborough program, but this is actually a spawning slick, so you get this really weird looking and very weird smelling, I have to say, pink slick on the surface of the water. So we can see when it's happened, even if we miss the actual event itself. So what we really wanted to do is figure out which environmental conditions predict coral mass spawning. And I put Q in there in brackets. That's what we were aiming at trying to get towards, but obviously it's a correlation. Like I said, we don't know much about this yet, so we're just starting to figure out what should our hypotheses be that we test further. People have looked at this before, but we've been a little bit limited in what we've been able to find out for a few reasons. The first is that often we have models that look for correlations with a particular environmental variable, but they only put in one variable. And for example here, this study included solar insolation and found quite a strong correlation, and so said this drives it. But they didn't test how relatively important that was. We have a lot of studies that have very few sites. So here I think we have 7 sites, and can't say that much about that, right? It's sort of a bit of a pattern, but it makes me nervous. And then we have other ones where we have quite a lot of things in there, but we only have it for one species, because it's really hard to get this information. So a bunch of people I've been working with took it upon themselves to get some real data on this stuff, and we've just heard about the importance of empirical data as well. So there's a couple of ways we can do this for corals to try and understand which months they're spawning in. The first is to just look for the spawning itself or for those weird spawning slicks. But the other thing we can do is we can actually go underwater and we can get with a hammer and chisel away at corals and get little bits of corals, and then we can look in them and we can see, so you might be able to see the little pink bits in there. We can see if they have eggs in there, and depending on the maturity of those eggs, we can tell if they're going to spawn in that month or the next month. So we can actually get data without having to be there on the specific night that they're spawning. So these amazing 11 co-authors on this study for 12 years have been collecting these data, and across all of these sites, which is 34 sites in the Indo-Pacific, they've got enough data to know for each month if they're spawning or not, which is pretty impressive. And this covers over 60,000 coral colonies of acropora species, which are the major habitat-forming corals. So the first thing we had to do was figure out when are they spawning. So there's no fancy modeling going on here, really. It's just what's going on. When are they spawning in different places? So we have a wheel here which shows you the different months of the year. And each black line shows you the months that corals are spawning in for a particular site. And then each site is ordered from the most northerly site in the center to the most southerly site on the outside. And the red dot is the month of peak spawning. So for most corals, they have the majority of their spawning happening in one particular month, but there's sort of dribbles either side of that. And what you can see is that there's a kind of a pattern coming out here already. We can see in the different hemispheres that they're sort of spawning at similar times to each other. So it suggests there maybe is some large-scale condition in the environment that's driving or cueing, maybe, that can predict this spawning. So we made a GLMM, so generalized linear mixed effects model, and we used monthly environmental data as predictors, so we were lucky to also have a co-author who works at NASA who can get hold of this kind of nice monthly data. It's quite hard to get otherwise. And then we looked at a bunch of variables that were hypothesized by other people to be important. So these are wind speed, current speed, timing of sunset, precipitation, temperature, and solar insolation or photosynthetically available radiation. And for those last two, we got the mean and the rate of change. So the rate of change is particularly something that people have suggested might be more important than just sort of static means. And yeah, we chucked in site and month, as you do, to make sure that we didn't have any trouble there. So the first one we did was we were trying to predict the month of spawning. So we weren't worried too much about peak in this one. This is just month, so any month that it's spawning in. And what we found was the strongest predictor variable is the change in sea surface temperature from one month to the next. So that was kind of cool. And we also had this effect of wind speed. So intermediate wind speeds, corals are more likely to spawn. So we're not sure if this is sort of a real thing or not, but it could be to do with the agitation of the water mixing or sort of separating the sperm and egg bundles so that they can then fertilize. We had a relatively high R squared, so 0.55 for marginal and 0.73 for conditional, and that the difference between those two suggests there's also quite a lot of stuff going on maybe that we're not capturing, that our monthly random effect is doing. So it'll be interesting to keep thinking about what that could be as well. When we look at peak spawning, we have sort of the same story in that change in sea surface temperature is again the most important predictive variable. And a slightly lower R squared, but not too bad, particularly in a macroecology session. So, so in conclusion, sea surface temperature rise across the seasons seems to be the most important predictor of coral mass spawning. And we think that this might be to do with sort of it cueing the corals to reproduce in these sort of Goldilocks conditions where we have a nice temperature for fertilization and for larval survival following that. But as I keep banging on about, because I think a lot about process, so I'm not that keen on correlations, but we have to start somewhere, right? Is this a cue or is it just a predictor? So that's something we're thinking about how to test now, and there's some people kind of working on this, and I'm not allowed to say anymore, but it's really awesome. So hopefully we can figure that out. And there's a couple of implications of this. The first one being that if that really is a cue, and the ultimate reason is to capture these Goldilocks conditions for survival of larvae afterwards, then what's going to happen under climate change? Are we going to get it decoupled, right? So if we think about the temperature changing over the year, and the fastest rise in sea surface temperature is when they're going to spawn, then under climate change we have maybe increase in sea surface temperature, but the regime is the same. So in theory they're going to spawn at the same time, but they're now going to be under different environmental conditions. Could be a bit of a problem, and it's maybe something we need to think about whether that's going to affect fertilization success and larval survival. And so that's particularly important for things like thinking about, are corals going to be able to recover from these horrific mass bleaching events that we've been seeing? The second implication is a much more simple one. Apparently, despite this massive mortality of corals and people repeatedly pointing out that maybe we don't want to dredge reefs and things like this, they're still going to keep doing it because they don't care. And so at least with these data, we can say, well, this month is really, really important. So can you at least stop dredging the reef, please, on this month? And then maybe the corals will have more chance of recovering and surviving. So it's beer o'clock, I guess. If you want to read more about this, it's in this paper, and the co-authors here are the ones that collected most of the data, so they're all awesome. Thanks. I don't know what the data says. I think there's a little bit of it, but to be honest, there's not much this year because everybody's been so focused on that. They've had the big task force going out, and they were doing the re-survey to look for recovery in November, which is the spawning month on the GBR. So I don't think there's too much data.
You didn't include lunar light intensity to predict the precise timing of spawning. Is there any particular reason why?
Yeah, so it's pretty likely that is really, really important, but that's important in determining the day. And so we're looking at trying to explain the month.
Is your data high enough spatial temporal resolution to predict the day?
No, unfortunately not. So some people do have data that is high enough resolution to do that, and that they're doing that kind of thing, looking at lunar cycles. But that tends to be in really small regions or just for one or two species. This is kind of like taking the macroecology approach to looking at this. Yeah.
Do you have any information about how the mass spawning would be affected with extreme climatic events? Because we are experiencing heat waves in the marine environment also. I mean, not only the variables would change, but the fluctuations would be quite impacted. Do you have any information on corals?
No, but I mean, it's really likely that it's going to have a big impact, right? Because whether it's got multiple points in the process where it could affect it. So it could affect it in terms of them getting enough nutrition to be able to start even producing the gametes, or it could affect them in terms of the queue now isn't working, or it might affect them in terms of the ultimate reason of why they're spawning at that time. So I'm sure there's probably a big effect of that stuff that we're nowhere near figuring that out yet.
Quite interesting to see that, yeah.
I think Sal's right. I think it is now beer o'clock.